Hello everyone and welcome back to IGCSE Science Academy. Today we are diving into part 3 of our unit on enzymes. In our previous sessions we defined what enzymes are. Today we are going to look at the how and why of their biological power. We will start with section 1 5.1.6 enzymes and active site, 5.1.7 specificity of enzymes, 5.1.8 effect of temperature and 5.1.9 effect of pH. By end of this lesson you will have a complete picture of why enzymes are the essential work horses of life. Let's get started with enzyme action. 5.1.6 that is enzyme action. Let's explain the components of enzymes one by one. Enzyme, a biological catalyst, speed of reactions without being used up. Substrate, the molecule that fits into the active site and is changed by the enzyme. Active site, a specific region on the enzyme with a unique 3D shape that binds to the substrate. Enzyme-substrate complex formed when the substrate binds to the active site held together briefly. Product, the new molecules released after the reaction. The substrate has been changed. 5.1.6 again, how enzymes work, the sequence. Now let's break down the actual mechanics of a reaction. To understand how an enzyme functions, we look at it as a four-step sequence. You can think of this like a specialized assembly line, or in many cases, a disassembly line. Step 1, that is the free enzyme. Step 1, we have a free enzyme. Notice that the active site is currently empty. The active site is a very specific shape waiting for its perfect match to arrive. Step 2, substrate approaches. In step 2, the substrate approaches. Because of random molecular movement, the substrate moves toward the active site. Remember, these two are complementary in shape, like a key moving toward a specific lock. Step 3, the enzyme substrate complex. Step 3 is the critical moment. The substrate fits perfectly into the active site, forming what we call the enzyme substrate complex. This is where the chemical bonds are either broken or formed. The enzyme is now actively lowering the activation energy required for the reaction to happen. Step 4, that is product released. Finally, in step 4, the reaction is complete, the products are released and they leave the active site, but look closely at the enzyme, it remains completely unchanged. We have discussed how enzyme works in our previous session. You can watch the complete video from there. So here is the key summary. The substrate fits into the active site, forms an enzyme substrate complex. The reaction occurs, substrate is converted into products. Products are released, enzyme is free to act again. It is not used up. The unchanged factor, this is the most common exam question topic, really emphasizes that the enzyme at step 4 is identical to enzyme at step 1. 5.1.7, moving on to a crucial characteristic of biology, enzyme specificity. If you take away only one thing from this slide, let it be this: each enzyme only works on one specific substrate. It's a specialist, not a journalist. So each enzymes only work on one specific substrate. This is called enzyme specificity. So why enzymes are specific? Each enzyme has an active site with a unique 3D shape. Only a substrate with a complementary shape can fit into the active site. If the shape does not match, no enzyme substrate complex can form. The reaction cannot take place. The enzyme is specific to that substrate. Now let's discuss about the lock and the key model. Look at the diagram on the right. This is the famous lock and key model. The enzyme is the lock, the substrate is the key. Just as your house key won't open your neighbor's front door, the wrong substrate, the red circle cannot trigger a reaction because it does not fit the lock's unique tumbler, or in this case, the active site. So in simple, the lock and key model states that an enzyme lock has a specific shape and only a particular substrate key fits into it. Now I want to draw your attention to the exam tip at the bottom. This is a classic trap in IGCSE paper. Never say the substrate has the same shape as the active site. If they had the same shape, they would just be two flat surfaces bumping into each other. You must use the word complementary. Think of it like piece of a puzzle. They aren't the same, but they are made specifically to fit together. Effects of temperature on enzyme activity. Now we are moving into the environmental factors that control enzymes. Temperature does not make things hot in biology. It changes how molecules behave and how they maintain their shape. First, kinetic energy, the speed up. Higher temperature, molecules move faster. More kinetic energy, separated enzymes collide more often. Look at our first box, kinetic energy. As we increase the temperature, we are essentially adding energy to this system. This causes the molecules to move faster. When the enzyme and the substrate move faster, they collide more often. It's simple math. More movement equals more frequent encounters. Second, that is effective collisions, the peak. Because of those frequent encounters, we get more effective collisions. These lead to more enzymes, substrate complexes forming every second, which causes the overall reaction rate to increase. This continues until we reach the optimum temperature, the perfect Goldilocks zone where the active site is at its more effective complementary fit. More collisions per second, more enzyme substrate complexes form, reaction rate increases. Shape and fit. At optimum temperature, active site shape is a perfect complementary fit for the substrate. Then denaturation, the breakdown. Too much heat, bonds in the enzyme break, active site shapes changes permanently, enzyme is denatured. But there is a tipping point. If we add too much heat, we hit the red box denaturation. High temperature causes the internal bonds holding the enzyme together to vibrate so violently that they break. When these bonds break, the 3D shape of the active site changes permanently. It's like melting a plastic keyhole. No matter how much you try, the key will never fit again. The enzyme is now denatured and can no longer catalyze the reaction. Remember, low temperature just make enzyme changing their shape forever. Always use the word denatured, never say the enzyme dies. Enzyme are not alive. But high temperature makes them stop by changing their shape forever. To bring everything we have discussed about temperature together, let's look at this summary table. This full picture view is exactly how you should structure your thoughts for an exam long answer questions. Temperature, the full picture. Temperature, what happens? Rate of reaction. So at low temperature such as 0 to 10 degrees centigrade, the kinetic energy is low. Molecules are moving like they are in slow motion. Because of this, we see very few collisions between the enzyme and the substrate. Even though the active site is perfectly intact and ready to work, the rate of reaction is very slow, simply because the players aren't meet often enough. Optimum, that is the 37 degrees centigrade in human. Then we hit the optimum temperature in human. This is typically around 37 degrees centigrade. Here we have the perfect balance. Maximum kinetic energy leading to the most frequent collisions combined with the enzyme's best physical shape and fit. This result in a maximum rate of reaction. High, for example, 60 degrees centigrade or 60 plus. What happens when we go too high, reaching 60 degrees centigrade or more, the active site shape changes. The substrate literally cannot bind anymore because the log has raped. This is denaturation. Even though the molecules are moving faster than ever, the rate of reaction drops to zero. It stops entirely. Key vocabulary. We have our 4 power words. Make sure you can define these. First is optimum temperature, the temperature at which enzyme activity is greatest. Denaturation, permanent changes in the 3D shape of the enzyme's active site due to high temperature. It cannot be reversed. Kinetic energy, energy of movement, higher temperature is equal to more kinetic energy. Effective collision, a collision between substrate and active site with enough energy to form an enzyme-substrate complex. 5.1.9 Effect of pH on enzyme activity. Just as enzymes have a preferred temperature, they are also extremely sensitive to their chemical surroundings. This brings us to the effect of pH. Whether an environment is acidic, neutral, or alkaline can literally change the chemistry of the enzyme itself. As you can see, the pH scale is mentioned here, which arises from 0 to 14. Let's start in the middle with the optimum pH, the green box. Enzyme is the ideal ionic environment. Active site has perfect complementary shape. Substrate fits perfectly. Maximum rate of reaction. Let's start in the middle with the optimum pH. In this ideal ionic environment, the enzyme is happy. The bonds are stable, meaning the active site has that perfect complementary shape we have been talking about. The substrate fits perfectly and we achieve the maximum rate of reaction. Now look what happens when the environment becomes too acidic. The red box, low pH. H positive ions interact with the enzyme. Bonds holding in the active site shape break. Active site shape changes. Substrate no longer fits. Denaturation. Too acidic. In a low pH environment, there is an excess of H positive ions. These ions interact with enzyme structure, causing the internal bond that hold the active site together to break. Once those bonds break, the active site shape changes, the substrate no longer fits, and just like with high heat, the enzyme is denatured. Now, to alkaline high pH, the blue box. The same thing happens on the opposite end of the scale. In a too alkaline environment, high pH OH negative ions interact with the enzyme. Again, those critical bonds break, the shape is lost, and the substrate is locked out. This is also denatured. OH negative ions interact with the enzyme. Bonds holding the active site shape break. Active site shape changes. Substrate no longer fits. Denaturation. The most important thing to remember here is that moving too far in either direction, too acidic or too alkaline, results in denaturation. Unlike temperature, where cold only slows things down, extreme pH levels on both ends of the scale will permanently change the enzyme's shape and stop the reaction entirely. Real examples and denaturation by pH. Real life enzyme examples, as we have already discussed in our previous videos. Enzyme pepsin, the location of this enzyme is stomach, optimum pH is pH 1 to 2, it's acidic and separate protein. So enzyme amylase, location mouth, small intestine, optimum pH 7, neutral, separate, starch. Enzyme trypsin, location small intestine, optimum pH 7 to 8, slightly alkaline and separate is protein. Enzyme catalase, location most body cells, optimum pH 7, neutral and substrate is hydrogen peroxide. So pH denaturation explained. Extreme pH means too many H positive ions, acid or OH negative alkali ions in the solution. These ions interact with the R group side chain of amino acids in the enzymes. The ionic bonds are hydrogen bonds holding the active site shape are broken. The active site changes shape. It is no longer complementary to substrate. So the enzyme is denatured. This change is permanent and irreversible. That brings us to the end of the unit enzymes. Today we have moved from the abstract lock and key theory into the gritty reality of how enzymes actually behave. Think about this: every breath you take, every bit of food you digest, and every single signal your brain sends depend on these proteins staying in their optimum zones. When you get a high fever, it's not just that you feel hot. Your enzymes are literally fighting to keep their shape so they can keep you alive. So here is a quick revision summary of enzymes part 3 and end of our unit enzymes, that is enzyme action, specificity, temperature and pH. We have seen that enzymes are very specific, their shape is their destiny. They are sensitive to their environment. Temperature and pH are the forces that hold an enzyme's 3D shape together. Once that shape is lost through denaturation, the biological machinery stops permanently. Now that you have mastered the theory, it's time to see how this appears on your exam. In our next video, we will be solving the topical past paper for enzymes. We will look at actual IGCSE questions, break down the marking scheme, and I will show you exactly how to phrase your answers to get full marks. Thank you so much for watching and learning with me today. If this helped you understand denaturation or pH better, do me a huge favor. Subscribe to the channel, like this video, comment below which enzyme you find most interesting. See you in the past paper session.